In the name of God, hello everybody. I'm Dr. Nourbakhsh. I'm a faculty member at the Department of Immunology at UMS, and the title of the class that I'll be teaching today is Development and Activation of B cells or humoral immunity. I'll be teaching three classes in this course. The first one is Development and Activation of B lymphocytes or humoral immunity, which is this class. The next class will be Development and Activation of T cells or cellular immunity, and that is the class after mucosal immunity class. And then finally, we'll be having a class about immunology of infectious diseases. I'm going to start from this very general schematic representation of immune system. You know from previous classes that immune system has two major parts, or it has two major arms. One part or one arm is called innate immune system, and that is the part which lacks memory and specificity. And the innate immune system is composed of physicochemical barriers and some cells, mostly phagocytes. And then we have another part or another arm, which is called adaptive immune system. And that is the part that has memory and specificity. And the adaptive immune system itself is composed of humoral and cellular parts. And the humoral part, B cells are responsible for, and the cellular part, T cells are responsible for it. So in this class, we're going to talk about humoral immune system, basically the story of B cells from the beginning to the end. And then in the next class, we'll be talking about T lymphocytes and so-called cellular immunity. We are going to learn three different things about B cells in this class. The first one is how B cells are produced in the bone marrow. That's where B cells are produced. What happens for B cells after they graduate from bone marrow? Basically how they get activated to produce antibodies. And then what happens for antibodies after they are produced, how they protect us from pathogens. Here I'm showing the same thing in a sort of a diagram. So this is the B cell story. The first part is B cell production and development in the bone marrow. The second part is B cell activation. And then the third part is B cell function, or basically function of antibodies. And if I want to show these things in terms of the anatomical location, the first part is going to happen in the bone marrow. So it's B cells in the bone marrow. The second part is B cells in so-called secondary lymphoid tissues, what happens for them there, and then what happens for antibodies which are produced by B cells. We will be having a similar story, three-part story for T cells in the next class. T cell development, T cell activation, and T cell function. Or if you want to talk about T cells in terms of their anatomical location, the first one is T cells in thymus, the second one in secondary lymphoid tissues, and then how they function. So, okay, so this class is basically B cell story. And I should warn you, this class is going to be a bit long. We used to have a separate class for B cell and T cell development, but then for this new program we had to decrease the number of classes, so the B cell development and T cell development part got combined with the activation and function part. But the good thing is that once we learn about B cell development and mechanisms for B cell development, T cell mechanisms of development would be very similar. So in the next class, it will be a bit shorter. In this class, we'll be talking about mechanisms of B cell development, which also applies to T cells. So the first part of B cell story, how B cells are produced in the bone marrow. Well, the most important aspect of B cell development in bone marrow is production of antibodies. Basically, antibodies are what distinguish B cells from other cells. And we know that well when we look at the surface of the B cell, antibodies are bound to the surface of the B cell, and they are called B cell receptors, and then afterwards they have to be secreted and released into body fluids, so that's a secretory or secreted form of antibodies. Why it's so sort of challenging to produce these antibodies? Because these antibodies are composed of four chains, light and heavy chains, and the terminal part or the N-terminal part of these chains have to be variable. Why does it have to be variable? Because this is the part which is going to bind to antigens. Or let's be more specific, it has to bind to different epitopes or antigenic shapes or determinants. And we have so many different antigenic shapes or determinants. So imagine that these are the so-called antigen binding part of antibodies. We have so many different shapes for these epitopes. And for the antibody to be able to bind to them, well, it has to have a complementary shape. Basically, antibodies have to have roughly the same number of shapes that antigenic determinants can have, and this is very challenging. So this is a huge problem. When antibodies were discovered in 50s or 60s, well, they had been discovered earlier, but I mean the structure was discovered. People were quite surprised. How is it possible for our bodies or for our immune system or B cells to produce antibodies which can bind to so many different antigenic determinants? How is it possible? Well, nobody exactly knows how many different uh, epitopes our immune system might face in a lifetime, but there are estimates. And well, we know one of the major things that our immune system has to deal with is pathogens. There are other things too, like neoplastic cells and cancer cells and stuff. But the most sort of challenging part are the pathogens, infectious agents, mostly microorganisms. And well, we will be talking about that in the next in the not in the next class, but the class after that. So how many different pathogens do we have, and those pathogens, how many different antigens can they have, and those antigens, how many different epitopes can they have? Nobody exactly knows, but there are rough estimates that there are thousands of different pathogens, thousands of different microorganisms which can cause disease. Those thousands of different microorganisms have millions of antigens. The smaller ones, like viruses, have a lower number of antigens. The bacteria have higher. Other ones, like fungi or helminths, have many more antigens. So there are millions of antigens. And each of those antigens have hundreds of epitopes. So we could have billions of epitopes, thousands of pathogens, millions of antigens, and billions of antigenic determinants or shapes. And our immune system has to be able to produce receptors, B cell receptors, which are antibodies, or T cell receptors with complementary shapes for those epitopes in order to protect us from them. How is it possible? It was very challenging for immunologists. This question was very challenging. Well, we don't know exactly the number, but there are estimates, as I mentioned. And the estimates for the number of epitopes or antigenic determinants is something on the order of 10 to 10 to 10 to 11, 10 billion to 100 billion different epitopes. That's the estimate for them. And our antibodies, well, our antibodies have to be able to bind to those many different forms, but considering that antibody is kind of a flexible molecule, each antibody can bind to epitopes with a similar shape. So the estimate for the number of antibodies which we require to protect us from those bad things is something between 10 to the 8 to 10 to the 9, 10 to the power of 8 to 10 to the power of 9, which is basically 100 million to a billion. That is the number of antibodies that we require. Some people have said that, okay, well, maybe 10 to the 7, 10 million different forms of antibodies is going to be sufficient, but even that is a very big number. So the question is how our B cells are able to produce those many different antibodies. Well, one simple strategy that comes to mind is to use different genes to produce different antibodies. We know that antibodies are proteins. When we look at a cell, like a lymphocyte or a keratinocyte or any other type of cell, it has a nucleus. That nucleus has chromosomes. Those chromosomes are DNA molecules, and those DNA molecules have different genes in them. The genes are parts or segments of the DNA molecule which are transcribed, and those RNAs which are transcribed from those genes, they can produce different proteins. So one strategy is that imagine that I require a sort of a molecular machinery that is composed of 3 different proteins, a red protein and a blue protein and a green protein. Well, one possibility is to have 3 different genes for them. And if I need, I don't know, 100 different molecules, protein molecules, well, I can have 100 different genes for them. So the strategy of simply using numerous genes to produce numerous proteins. And we have a system in our body which does that, and that's the olfactory system. Olfactory system, like immune system, has to produce receptors for different molecules, and those molecules, they are not antigens, but they are molecules that have a smell, basically odorants. So our olfactory system in humans does possess almost 500 different forms of olfactory receptors, and it uses roughly the same number of genes to produce those receptors. So if you look at our genome, the genes for olfactory receptors are spread around everywhere, and there are many of them, hundreds of them, and they produce olfactory receptors for us. In other animals like rodents or dogs, which have a much better sense of a smell, the number of olfactory receptors and the corresponding genes is something around 1,000. So that's one strategy that has worked well for our olfactory system. Does it work for our immune system? Can we assign numerous genes to produce antibodies for us? Well, if we look at the number of antibodies which we require, and I mentioned that the lowest estimates are 10 million different forms of antibodies to be to enable us to protect ourselves against pathogens. Well, we figure out that that strategy is not going to work for immune system, because the total number of protein coding genes that we have in our genome is something around 20,000. If I want to use the strategy of numerous genes for numerous proteins to produce antibodies, this genome would simply not have enough space. We needed to have a much, much bigger genome, almost 400 times bigger, and then assign the entire protein coding capability of that genome for production of antibodies. To be able to produce these many antibodies. So it's not going to work for our immune system. Our immune system actually has come up with some other mechanisms, which I think they are some of the most fascinating things which exist in biology. And well, we'll see if you agree with me at the end of this class or not. And I'm going to tell you about those mechanisms. Those, or let me say, call them strategies for production of numerous forms of antibodies. In order to explain these strategies, I'm going to use a metaphor which does not belong to biology, does not come from biology, but it comes from computer science. And it's a metaphor which is good for the millennial, so-called millennial generation. What is the millennial generation? Well, it's your generation. I guess most of you guys belong to the so-called millennial generation, and it has a definition like people who's been born between, I think, 1985 to 2005. So that is the so-called Generation Y, or the millennial generation. And this is the generation that is very, very familiar with information technology and spends a lot of time with computers and monitors and cell phones and stuff like that. So that metaphor is going to be suitable for you guys. I can explain the mechanisms that I'm going to explain without using this metaphor, but I think it helps. So bear with me for a few minutes so that I can explain it, and then we'll get back to the immune system. So what is the metaphor? That's the metaphor of combining basic colors to generate many different colors. So that's basically the metaphor that we're going to use. So this is the first generation of monitors. You might have seen these monitors. These were the first generation of computer displays or monitors that became commercially available, I think, in 1950s. They were called monochromic or monochromatic monitors because the pixels that they had could be either on or off, and of course you could not show any colored pictures in these monitors. The next generation of monitors were the monitors whose pixels could produce blue and red and green at the same time. If all of these three colors were on, you would be getting a completely white pixel. If they were off, you would be getting a black pixel, and then you could have different combinations. So it was a big development compared to the monochromic monitors, but still was not very good. If you wanted to show like this picture of parrots, this would be the thing that you would be seeing. So further development was needed. How was it possible to make pixels which could show many different colors, like the currents that we require to produce or to show or to display this impressionistic picture, for example? Well, would it be necessary to produce pixels which could show each of those particular colors separately, or that it was possible to do it somehow differently? Well, they started generating pixels which could show red, green, or blue, but not pure red, green, or blue, but different shades of green, red, and blue. So if I have a pixel which can have 10 different shades of green, 10 different shades of red, and 10 different shades of blue, and then combine these different shades, I could be having 10 times 10 times 10 different combinations, and that would give me a pixel which was able to produce 1,000 different colors. So by combining three basic colors, we know that we can have different colors. By having different shades of those colors and then combining them, we can have many, many different combinations. So this gave rise to the so-called imaginary color space that people called it RGB color space. And well, this color space, or this cube, got bigger and bigger by including more different shades of these colors. If we look at the computer monitors that we have today, or the cell phone monitors that we have, if we go and open one of these softwares that has a so-called color palette, we see that each of these colors has 256 different shades, and then you combine these different shades and you get millions, roughly 16 or 17 million different combinations. This is called combinatorial diversity, a diversity which is produced by combining different basic elements, and those elements have to have different levels. In this case, for the colors, they have to have different shades, or in the case of antibodies, as we will see, they have to have different sort of versions. So a bit of a reminder from molecular biology class before we see how combinatorial diversity is implemented during the evolution of immune system. We know that genes are part of DNA molecules which are transcribed by the RNA polymerase enzymes, and RNA is generated. In the beginning, it's called an immature RNA. It has exons and introns. The introns have to come out during a process which is called splicing, and then you will have a mature RNA molecule. In this process of splicing, we have a limited possibility for diversity, and it's through so-called alternative splicing. We can have like different exons binding to each other. So this is one possibility for generating different proteins from a single DNA molecule, but it's very, very limited. Like if I have 4 different exons, I don't know, I can come up with maybe 4 or 5 different splice variants. So it's good, but it's not sufficient to produce many, many different forms of proteins that we require, for instance, for antibodies. So the genes that we're looking at here, they're called the so-called continuous genes. Continuous genes, even though they have different exons and introns, but these exons can come next to each other at the RNA level, and the RNA polymerase is ready to transcribe the gene from the get-go. So it transcribes the entire thing, and then an RNA is produced, and that RNA undergoes a splicing. We have another type of genes, which are kind of the minority in our genome. They are called discontinuous genes, and these are genes which have multiple fragments. Pay attention, these fragments are very different from exons and introns. So each of these fragments can have multiple exons and introns. But these fragments are located in different parts of the genome, usually on the same chromosome, but they are far away from each other. And this precludes the RNA polymerase from transcribing them. For these genes to be transcribed, they need to undergo a process which is called recombination. So the parts in between have to be cut out, and then the parts next to them have to be brought next to each other, and then we will have a so-called continuous gene, and then it can be transcribed. So this recombination is something which happens at the DNA level. It's very different from splicing, which happens at the RNA level. So keep that in mind. So when we look at the parts of the genome which are going to produce antibodies, and for an antibody chain to be produced, we have to have a variable part, and then we have to have a constant part. The problem that we have now is the variable part, because that is the part that has to have many different shapes. So here I'm showing you a DNA molecule which has sort of, my mouse is talking here. Okay, so the variable part and then the constant part. The funny thing is, when you look at the part which is going to produce the variable region, you will see that in the B cell, in the beginning of its development, before it's undergone its development, the gene for production of the variable part is a discontinuous gene. So it has multiple fragments, and there are 3 different fragments. And this is the same in all of the cells in our body, by the way. So DNA molecule is the same everywhere, including the so-called hematopoietic stem cells, which are going to produce B cells. So this is discontinuous. It has to undergo recombination before it can be transcribed. But the interesting thing is that when you look at the part which we call it, we've called it here fragment 1, or fragment 2, or fragment 3, you will see that each of these fragments has many different versions. So it's not a single fragment 1. We have tens of fragment 1s that I've called them fragment 1 A, B, C in here. So these fragment 1s, these different versions of fragment 1 are overall, they have a similar sequence, but there are also tiny differences. So for fragment 2, again, we have multiple different versions, A, B, C, D, all the way to the end. So overall, they're similar, but they have tiny differences. And then for fragment 3, and we will see that these fragments have, they've given them names. So for a variable protein, or the variable part of the heavy chain, which is VH, or variable part of the light chain, which is VL, to be produced, we need one of these fragment 1s, one of these fragment 2s, and one of these fragment 3s. So recombination has to happen between one of these fragment 1s, doesn't matter which one of these fragment 2s, and one of these fragment 3s. So these are different versions, similar to different shades of those 3 basic colors that we have for the pixels for the monitors. So this is for the heavy chain. Here I am showing you the antigen binding part of the antibody. So I'm using green color to show you the light chain, or the VL, variable part of the light chain, and the blue one for the variable region of the heavy chain. So the variable region of light chain, the gene for it has 2 fragments, and each of those fragments have multiple versions. And the variable region of the heavy chain is composed of, the gene for it is composed of 3 fragments, and I've called them, as I showed you in the previous slide, fragment 1, fragment 2, and fragment 3. In order to make them sort of sort of nicer in terms of their names, people have given them names instead of fragment 1, 2, and 3. They've been called, fragment 1s have been called the V, the fragment 2 has been called J, and fragment 3 has been called D. This is for variable joining and diversity. But these names are kind of misleading. All of these fragments are going to produce the variable part of the antibody, but these are simply names. So we call them V fragment and J fragments. For the VL, variable part of the light chain, it's called V and J, 2 fragments. And then we have V, D, and J for the VH, or the variable part of the heavy chain. So for the rest of this part of this class, I'm going to focus on the heavy chain part, which has 3 fragments. So slides that I'll be showing you basically are this part. So what happens when a B cell starts its development in the bone marrow? Well, we have the V fragments, multiple versions of V, D fragments, multiple versions of D, and J fragments, multiple versions of J. When a B cell starts development, the recombination process takes place in these V, D, and J fragments. What happens is that some enzymes, whose action is to perform recombination at the DNA molecule, come and take one of these V's, completely randomly, one of these D's and one of these J's, and put them next to each other. Basically, they cut the part in between, and then they bring the parts on the sides to sort of, and join them together. For instance, in this particular combination that I'm showing you, V2 is joined by D3 is joined by J2. So this might happen in one lymphocyte. In the next lymphocyte, another V joins another D, joins another J. And in the next lymphocyte, another V, another D, another J, and so on and so forth. So here you're seeing that we can have many different combinations. Imagine that I have 100 different V fragments, 10 different D fragments, and 10 different J fragments. Overall, I have 100 plus 10 plus 10, which is 120 different fragments. But they can combine in 10,000 different ways. So they get multiplied 100 times 10 times 10. So they can give us 10,000 different so-called VDJ forms. And that is how combinatorial diversity takes place at the level of genes for B lymphocytes. So this is one combination, another combination, and we can have like 10,000 different combinations. I'll tell you about the exact figures for the number of these fragments, but roughly, like, imagine that 100 times 10 times 10 can give us 10,000 different fragments. 10,000 different combinations. So enzymes that do this work, they are called recombinases. When we have two major recombinases in lymphocytes, one of them is called recombinase number 1, or recombination activating gene 1, or RAG1. The other one is called recombination activating gene 2, or RAG2. So these are the enzymes which are responsible for recombination. And in order for the recombination to take place, these enzymes have to be able to recognize where to cut and where to paste. And that is what happens by the so-called recombination signal sequences. So what are these sequences? Well, if I zoom in on this DNA sequence, imagine that I'm zooming on this DNA sequence on the V3 and maybe D2, and I'm making it a bit bigger. When we look at the sequence for these genes, we will see that, okay, so there is a sequence for the V3 itself. This is the part that is going to produce some protein, and then D2, again, this is the part which is going to produce some protein. But on the right side of the V fragments and on the left side of the D fragments, we have some particular tiny sequences, some heptamers and some newnomers, some 7 nucleotides and some 9 nucleotides, which are located on the right side of the V regions, and exactly the same thing on the left side of the D regions, but in opposite direction. And these are called recombination signal sequences. These are parts which tell the recombinases where to cut and where not to cut. So a recombinase always looks for these 7 mers and 9 mers on the left side, and then 7 mers and 9 mers on the right side, and then brings them together, cuts the region in between, and then attaches or joins the parts on the outside. So if I want to show you this a bit better, this is a better picture. Again, a DNA molecule. We have the V fragments, D fragments, and J fragments. I've kind of made them a bit bigger in here, but this is basically a DNA molecule. For a recombinase to do its work, it always looks for these 7s and 9s. So it takes 179 from the left side, 179 from the right side, cuts the region in between, and joins them. Well, the way that I'm showing in here, sort of disappears and then joins. But in reality, it sort of loops out and then cuts the region in between and then attaches the parts next to them. So a D comes next to a J, and then another recombinase comes. Again, it recognizes 179 on the left, 179 on the right, cuts the region in between, and joins them. And this process of recombination is called VDJ recombination. And this is completely random. Like each of these V's could be selected, each of these D's could be selected, or each of these J's could be selected. But it has to be selected and cut exactly on the parts next to the 7 9s, so that it guides these 7 9s, or these recombination signal sequences guide the enzyme where to cut and where not to cut. That is how the RAG enzymes know where to basically perform the recombination. So this is called the VDJ recombination. In the beginning, D comes next to J, and then V comes next to DJ, and then it will give rise to the continuous gene which is required for production of the variable part of the heavy chain. The same thing happens for light chains, but in the genes for light chains, we don't have the D segment, or we don't have the D fragment, it's only V and J. And then we have the so-called VJ recombination. So this is the way that this recombination takes place. Okay, let's look at the chromosomes where these things happen. So we have 3 different chromosomes which are involved in this process. Chromosome 2, 14, and 22. Well, this is kind of important for those of you which are going to specialize in hematology and oncology in future, because these are the chromosomes which get involved in leukemias or lymphomas. So keep that in mind. This is important for those of you who might become hematologists or oncologists. So chromosome 14 is the one that is responsible for generating the heavy chain, and chromosomes 2 and 22 are responsible for generating the light chains. And we know that light chains, there are two types of them, kappa and lambda. So 2 is responsible for generating the kappa light chain, and 22 for the lambda light chain. So if I zoom on the heavy chain part, there is a tiny part close to the telomere of chromosome 14. This is the factory for producing the VDJs, and you see that there is a small region in here that gives rise to all those different VDJ fragments which we require for production of the variable part of heavy chain. So you don't need to assign the entire chromosome or a huge part of genome to produce, like what olfactory system does, to produce these different forms of antibodies. A tiny region, but with a very interesting and clever and fascinating strategy, which is DNA recombination. So let's look at the numbers. Here I'm going to use something similar to what computer scientists use for showing the different combinations of colors. There we had the RGB space for red, green, and blue. Here we have the VDJ space for variable joining and diversity for the heavy chain. So the number of V chains, roughly 100. The number of the versions of the V fragments, roughly 100. For D, the number is roughly 27, and then for J's it's roughly 6. And I say roughly because different studies have shown slightly higher or lower numbers. So if we multiply 100 by 27 by 6, we get sort of a rectangular cube in here, and the number of combinations is 16,000. So that is the number that we get by multiplying V's and D's and J's. So that is for the heavy chain. For the light chain, we don't have the D fragment, so it's only V and J, roughly 35 for V, and 5 for J. So it's sort of the previous one is a 3-dimensional space. This is a two-dimensional space, and the number is 175 when you multiply them. Each of these heavy chains can get bound to each of these light chains. So the number up in here is going to get multiplied by the number in here, and it will give rise to roughly 3 million different combinations, which is a very, very good number. So it was a very efficient strategy, the so-called VDJ recombination, which is basically responsible for combinatorial diversity for antibody molecules. So it gives us roughly 3 million different antibodies. But we still need a higher number. If you recall, the lowest estimates for antibodies that we require was 10 to the 7, which is 10 million. And actually, most people say that, no, okay, 10 million is not nearly enough. We need at least 100 million different antibodies. And here we're getting only 3 million, which means that we need another strategy. The next strategy that I'm going to tell you is called junctional diversity. But before I tell you that, I need to mention something. I said that this process of recombination is a completely random process. So there is no sort of supervision in the bone marrow checking which lymphocyte is producing which combination. So it's a completely random process. And if it's a completely random process, one thought that comes to mind is that, well, who knows, maybe I'm missing some of these combinations. Who can be sure that I have all of the combinations which are required for protection against pathogens? Let me get out of this slide for a second. So let me type in something in here. So imagine that for me to be able to protect myself against let me get rid of this, against a particular pathogen like influenza virus or Ebola virus, or God forbid for SARS-CoV-2, I need to have an antibody which has a particular heavy chain, or let me call it VH. And that heavy chain requires, let's say, V67 next to D13 next to J4. So this is what I need for the VH part of that antibody. And then for VL, for that particular antibody, which is going to protect me, hopefully, against that particular pathogen, I'm going to need V15 next to J8. Okay, so this is just I'm just coming up with some numbers. So how do I know that I have this particular antibody in my so-called antibody repertoire? How to be, let me type it in here, how to be sure that I have this very, very, very particular antibody in my B cell repertoire. So let's learn a word in here. So B cell repertoire, it's a French word. A repertoire is basically a set of things which have similarities but some differences. So it's called the B cell repertoire, the entire set of B cells that I have. How can I be sure that I have this? Maybe I'm missing this particular combination, or maybe you're missing some other combination, or your friend is missing some other combination. So this is a thought that comes to mind, and actually we might say that, yeah, well, it's a random process, so I might be missing some combinations, and maybe that is why some people are more susceptible to some particular disease. So here's the question: are we really missing this combination or some particular combination? Considering that this process is a completely random process, so I might be missing something. Well, the answer is that no, I'm not missing anything, and you're not missing anything. We can be confident that we do have all these possible combinations in our B cell repertoire. So my B cell repertoire is not missing anything. How can I be so sure? Considering that, well, nobody's checking the B cells in the bone marrow, and it's a completely random process. How can we be sure? Well, in the classes that we're physically present, I usually ask the students to give me some ideas about this problem. And well, those classes are a bit more interactive, but now I'm recording something, so it might be nice for you guys to pause and ponder for a few seconds about this question. How can we be sure that we have all possible different combinations for our B cell receptors, considering that it's a random process? And the same story for T cells. Well, I'm going to tell you the answer, and the answer is related to the probability mathematics or statistics. So, well, there is this story about a teacher that goes to a class one day and he's going to teach something about probability. He goes to one of the students and asks, well, could you tell me your birth date? And that student says that, okay, well, why do you want me to tell you the birth date? Well, how is it related to, could you tell me your birth date? So I'm using this as a black birth date. How is it related to the probabilities and stuff? Well, the teacher insists, and he or she says that, oh, okay, so I was born, and the year is not important for us. Oops, sorry. The year is not important for us in here. It's just the date itself. So he says or she says that maybe 6th of September. Okay, 6th of September. That is the day in the year that I was born. I don't know, in 1980 or some other year. So the teacher says with a surprised face that how is it possible? How is it possible? How is it possible for somebody to be born on 6th of September? The probability is very low. The probability is extremely low, is very low. What is the probability for somebody to be born on 6th of September is 1 over 365. And it's less than, I don't know, 0.3% or something. So it's a very low probability for somebody to be born on a particular day in a year. However, we never feel surprised if somebody's been born on 6th of September, the 1st day of January, or the 10th day of August. Why? Because the number of children which are born every year is much higher than the number of possibilities which exists for the birth dates. So we have only 365 possibilities in here. But every year in a city or in a country, millions of children are born, and well, they have to fill all of these possibilities, so they will fill them easily. So all of these possibilities are met, and not only all of these possibilities are met, we don't see any particular day that nobody's been born. At every particular day, we have hundreds or even thousands of children which are born in a single country. Have those children been coordinating with each other? Like if you look at 6th of September, I'm sure that in every country that you look at, there are hundreds of children whose birth date is 6th of September. How has it happened? Have they been coordinating or arranging with each other? Let's be born on 6th of September, or I don't know, the first day of January. No, it's been a completely random process. But the number of events, the number of events has been much, much, much bigger than the number of possibilities. And this is the basic mathematics for it. For those of you who might be a bit more interested in this, this is related to a concept in mathematics which is called the pigeonhole principle. Pigeonhole principle. And if you want to learn about it, you can read about it. So it deals with events compared to the number of possibilities that those events can have. And then if the number of events is much higher than the number of possibilities, all of those possibilities are met. So this is what happens for B lymphocytes. Every day in a single individual, billions of B cells are produced. And these billions of B cells have only roughly 3 million possibilities to fill for their VDJ and VJ recombinations. We can be sure that all of these possibilities are met. And not only that, I'm not missing anything. For each of these individual possibilities, like this particular antibody in here, I have thousands or even millions of B cells with this very specific VDJ and VJ recombination. Exactly the same thing, like children that have been born on exactly the same day. Why? Because, well, billions of B cells and only a few million of possibilities for antibodies. Well, each of those possibilities are met, and for each of those particular combinations, we have many, many different B cells. So that's what happens. Okay, so that sets us for talking about the next strategy, and I'm going through this a bit fast because we need to talk about activation and function of B cells. So imagine that I have a particular combination B13 next to D2 next to J4 for heavy chain of an antibody. The interesting thing is that, well, when the RAG enzymes are going to do this process, they have to be a bit of a cutting and pasting. So they have to cut the region in between, and then they have to sort of join these separated fragments. So this gives rise to a few milliseconds of time for this next strategy to implement itself. What is that strategy? We have DNA fragments in here, and these DNA fragments have been separated by the RAG enzymes. They've been cut, and it takes a few, I don't know, milliseconds for them to rejoin. We have a particular group of enzymes which are very fond of free DNA ends. They like those free DNA ends. They are called exonucleases. And these exonucleases, whenever they see that there is a DNA end which is free, they get very, very happy and they start cutting those DNA ends. So, and this happens in those few milliseconds between cutting and pasting with the RAG enzymes. So they start cutting some nucleotides from here. So imagine that like this particular exonuclease start eating, it cuts out some of these nucleotides. How many? Well, we don't know. It could be completely random. Depends on whether there is an exonuclease there and how much time that particular exonuclease has. It could be nothing, or maybe 1 or 2 or 3 or 10 or I don't know, 20 different nucleotides could be cut. The same story happens for the free ends of the D and free ends of the J. So this is what happens here. And keep in mind that this particular V13, D2, and J4, it doesn't exist in a single lymphocyte. It exists in millions of lymphocytes because many of those lymphocytes have this very, very particular combination as we talked about in terms of probability and statistics. And then again, we have another group of enzymes which are called TDTs, terminal DNA transferases, terminal deoxynucleotidyl transferases. And the action of these TDTs is completely opposite to exonucleases. Whenever they find a free DNA end, they start adding nucleotides, completely random nucleotides. Like this particular TDT might be adding this particular nucleotides here. This one might be adding this one, completely random depending on which nucleotides are available. And then they add these different things. And after these exonucleases and the TDTs have sort of played with those free DNA ends, then the RAG enzymes attach or join these ends to each other. This is a process which has actually the molecular detail for this process much more than what I'm showing you here. But we're trying just to get the concept, so the details are not very important. There are many more enzymes involved in this process, but I'm showing you only two of them. So the combined effect of removal and adding of nucleotides gives rise to many V13D2J4s, which are similar in terms of VD and J, but the region in between is very different. And each of these things happens in one particular B cell. We have numerous B cells with this very particular VD and J combination. So in terms of VD and J, they are the same, but the region in between, which I'm showing with these different colors, is going to be very different. And this region could be quite big, actually. So I'm showing it's sort of small, but it could be hundreds or tens or hundreds of nucleotides. So this particular VDJ has this particular junction between B and D and D and J. This another lymphocyte, again the same V, the same D, and the same J, but the region in between is different, and then another lymphocyte and another lymphocyte, and this is a process which is called junctional diversity. So in contrast to the previous one, which it was diversity due to different combinations, this is the diversity due to differences in nucleotides in the junctions between V, D, and J's. Okay, so combinatorial diversity times multiplied by junctional diversity gives rise to much higher number of antibodies, roughly 10 to 10. So this is actually even higher than the number that we expected. The number that we needed was almost 10 to the 7 or 10 to the 8. But in reality, actually, if you look at the number of antibodies that we have, most of these antibodies that are produced, they are not good options because it's a random process. So a lot of antibodies which are produced are bad antibodies. They're antibodies which can bind to our own antigens. And those B cells are going to be deleted. They are not allowed to mature. So finally, in a mature person, the total number of B cells that you see in the B cell repertoire is roughly 10 to 7 or 10 to the 8, because a lot of the antibodies which we're calculating in here are not going to realize. All right, what happens next? Well, after the VH is produced, and similarly the VL, then we need to have the complete antibody molecule. So this was only the variable part. We also need the constant part. So here I'm showing the DNA again. After the VDJ recombination has taken place, we have different classes of antibodies. Those different classes or isotypes of antibodies have different constant regions. For a B cell to mature, we need to have, in the beginning, we need to have two antibody molecules of two classes. Those are IgM and IgD at the surface of B cell. How is this going to happen? Well, an RNA molecule is made. So following the completion of the so-called VDJ recombination, transcription can get activated. And then an RNA molecule is produced. So this RNA has the VDJ on one part of it, and then M and D on the other parts. And then these are dealt with like exons in an immature RNA molecule. So splicing takes place. And splicing can give rise to different basically mature RNAs or different transcript variants. So in one form, the VDJ comes next to the M, and in the other form or in the other transcript variant, VDJ comes next to D. And this one produces the IgM molecule, and this one produces the IgD molecule. Two different antibodies with two different classes, but the variable part of those antibodies are the same. So this is the beginning of generation of antibody molecules. And this happens in the bone marrow. For the rest of the antibodies to be produced, and this happens, as I'll show you, after the B cell is graduated from the bone marrow, we need again to have a recombination at the DNA level. So again at the DNA level, a recombination process needs to get activated, and then VDJ needs to come next to, I don't know, one of the G fragments. You know that we have 4 different subclasses of IgG. In humans, there are 1, 2, 3, 4. The one that I'm showing in here, it's 1, 2A, 2B, and 3. This is for mice. But for humans, it's 1, 2, 3, 4. So the VDJ comes to next to one of the G's or one of the other ones. Like if I need IgG, this one will happen. If I need IgE, this one will happen. If I need IgA, the next one will happen. What does determine which of these antibodies we require? We'll see it in the next part of the class. It's the signal from T lymphocytes, which tell tells B lymphocytes which of these antibodies they need to produce, and we'll see it in the next part of the class. But I'm going to wrap up this part because it's already getting too long. So different antibodies from different classes are produced. Pay attention that these different classes of antibodies have exactly the same VDJ on the heavy chain and VJ on the light chain. So in terms of antigenic specificity, they are similar, but the class of the antibody is different. For instance, we have an antigen in a virus, like in influenza virus, we have an antigen which is named hemagglutinin, it's HA. We can have different B cells which produce IgG anti-hemagglutinin or IgE anti-hemagglutinin or IgA anti-hemagglutinin. So the variable part for these are similar, but the class of the antibody is different. And we'll see that those classes are important. Functions of those classes are important. So we'll see it in the third part of the class. All right, so this ends the story of B cell development. I'm just showing you some cells. We were talking about molecules and DNA up to now, but let's put these molecules inside cells. B lymphocytes originate from a hematopoietic stem cell. For a B cell to become mature, it has to undergo some steps, the pro-B, the pre-B, immature B, and mature B stages. Those things that we talked about, the DNA, we talked at the level of DNA, they are completely related with these 4 different stages. I'm showing you here what happens for each of these stages at the DNA level, at the cell surface, in terms of antibody molecules, and then the class of that antibody. So the cell that we call it pro-B, the recombinase enzymes are getting activated, but they have not yet done anything. So at the level of DNA, we still have the original DNA. It's called the germline DNA, and there are no antibody molecules at the surface of the cell. In the pre-B cell, the heavy chain undergoes recombination. So the heavy chain undergoes recombination first, and then that heavy chain produces and sort of an incomplete antibody molecule, which is composed of only 2 heavy chains. So they appear at the surface of the cell. And there is a tiny molecule, because heavy chains don't like to appear at the surface of cell by themselves. They kind of bind the molecule which looks like a light chain, but it's not really a light chain, and they appear at the cell surface. So it's an incomplete antibody molecule. In the immature B cell, in addition to the heavy chain, which has already undergone recombination, light chain also undergoes recombination. Then we have a complete antibody molecule of the IgM class at the cell surface. So this is a complete IgM molecule. And by the way, this is a monomeric IgM molecule. So it gets polymerized. It becomes a pentamer IgM after it's secreted. But at the surface of the cell, it's a single monomeric IgM. This is called immature B cell. Why immature? Because B cells require a second class of antibody before they can be labeled mature. So they need to produce IgD as well before we can call them mature. So the joke with the medical students is that these B cells, like you guys, need to get their MD degree, so they have to have an M and D before they get sort of the license to practice, to work. And that happens after the B cell is released from the bone marrow. Okay, so this was the first part of B cell story, the B cell development. The next 2 parts are B cell activation and B cell function. So I'm going to save this video file up to this moment, and then in the next video file, the second part of this class, I'll be talking about B cell activation. All right, so let me get out of here and save this.